So we're moving on the dogs and cats, and the goal of this lecture is essentially just going over an overview of what we know, or better, what we do not know about the pathogenesis of food allergy in dogs. And I'm specifically focusing on dogs just because in dogs we know a little, in cats we know essentially nothing. So, so let's focus on dogs, at least we know a little bit of what happened. And obviously the pathogenesis is important, but part of this lecture I'm going to focus on allergens, and what are the common allergens, what are the characteristics of allergens, and how important they are in our practice. So, without offending anybody in this audience, I'm sure that everybody is fully aware of the distinction between the non-immunologic, the food intolerance and food allergy. So just a reminder for the people online maybe that that food intolerance and characterized by non-immunological mechanisms, difference with the allergic reaction, which are characterized by the immunological mechanism triggering the disease. Now, in people more than in dogs, the characterization of the immune response behind the different type of food allergy manifestation is much more clear than what we have in dogs. So in this graphic, you can see like differentiated between non-IgE mediated, IgE mediated. Well, it's very easy on a graph. It's very difficult on the practice to try to figure out which one we're actually dealing with on a daily basis. And again, I'm a dermatologist, so probably the type of population that I see in my practice may be a little bit different for what you guys see in your practice as a clinical manifestation of food allergy, and of course, the correlation with atopic dermatitis that Dr. O'Mahony was talking about. Now, food allergy is characterized by a chronic non-seasonal disease. That's extremely important for my patient. And obviously, we have clinical signs which are compatible or completely overlapping with atopic dermatitis, with very few exceptions. So most of our patients do have skin issues, but they also have a variety of other symptoms like GI problems, and most of likely those are the one that you guys see on the regular basis. So for my slide of population is the dog comes, goes, scratches, and by the way, has a GI problems. Okay, so that's my type of patient. And so most of our patients will have itch, which is characterized by chronic, non-seasonal, generalized in a sense, and it may have also some GI problems specifically associated with increasing number of flatulence, increasing number of defecation, diarrhea, vomiting. Most of the times, those signs are very subtle for my patient. Matter of fact, if we don't ask specifically what the dogs have, most of the time the answer will be, my dog does not have any GI problems. Absolutely none. And I had plenty of patients that come when they're puppies, and I have the owners like, how is Fluffy pooping? And I was like, oh, it's fine, it's normal. It's like, okay, can you pick it up? Oh, no, but that's him. It's like, okay, that's not him. There is a problem. And then you start to change the diet, and it starts to get better. Of course, in this case, it's not just the diet-responsive diarrhea that my friends in internal medicine would deal with. Those guys will also come with a chronic recurrent otitis with chronic itch in those cases. They also may have other clinical signs. Those are much more rare and essentially anecdotal in most of these cases, but they may have respiratory problems. They can have rhinitis, conjunctivitis, anorexia. There have been even reported cases of glomerulonephritis associated with food allergy, or even perianal fistula in some cases associated with a food allergy. Very uncommonly, but has been reported. So, how common is this disease? Well, it has nothing to do with, in terms of frequency associated like with diseases like atopic dermatitis, which is much more common in the canine population when we're talking about skin diseases. But this is a review that they put together a few years ago. Dr. Ralph Mueller and Thierry Olivry from Munich and North Carolina State University. They put it together with a series of cats. So these reviews to essentially critical appraisal reviews to put together like different incidents or clinical science presentation and type of rechallenge and time for diagnosis, how to make a diagnosis of food allergy in our patient. And essentially, depending on what is the slide of population we're talking about, it can be as low as 1% of any disease in terms of dogs that show up at the general practitioners or as they bring it to visit, up to more than 20% of dogs with associated atopic dermatitis or with a pruritic skin disease. So it can be quite common. In practice, what we see generally is any dogs, we have about like 30% of dogs with atopic dermatitis may have food allergy component, which goes very well with what has been shown in this review. The breeds predisposition, we do see some breed predisposition, mainly are retrievers, and German Shepherd or West Island, which are actually also the breed that are highly predisposed with atopic dermatitis. So with Dr. O'Mahony, we saw about the atopic march, how in people everything starts with the skin and atopic dermatitis, and then it starts to evolve with acquiring a food allergy that goes with rhinitis, and that goes with asthma, which is a beautiful model that they have in human of the atopic march. Well, that doesn't really happen in our patient. Our dogs do not get asthma very commonly. They do not get allergic rhinitis very commonly. They do get atopic conjunctivitis, specifically according to our ophthalmologist, which anytime there's a running eyes, like, oh, it's allergy, go to dermatology. But we do have the majority of our patients will develop just like a skin type of problems. And that's what we see also with food. Age of onset is one of the very few diseases that it shows up mainly in early life. So we have up to 40% of our food allergy patients showed up with the onset of disease before 1 year of age. And then nothing kind of happen until they get old. And then you start to have another wave of these dogs developing food allergy. Obviously, when we talk about age, yesterday I was interested when there was the thing like old dog above 8 years of age. I guess I never seen under that perspective, because for me, an 8 years old Chihuahua, I'm not sure that I would consider old, considering those dogs never die, you know. An 8 years old Grand Dane, yeah, absolutely, those are ancient dogs. But definitely like you have this trend in the older age. And like as you can see from this review, anything can go between like 6 months of age up to 14 years of age, which is characterizing like this type of dogs, the type of population that we see every single time in our practice. So again, clinical signs. I try to don't put like gross pictures. I know many people hate dermatology because of very gross pictures, but you know, those are pretty decent, I guess. So we have like a very scratchy, itchy dog with overimpression of, I already see faces around, with some malassezia or cutaneous bacterial dysbiosis, some abdominal erythema, and then obviously this wonderful nasty otitis that has been going on forever and ever with this dog that may sleep with the owner, no judgment. But we do have also, again, like I said, gastrointestinal problems. Most of the time we do have diarrhea, but again, we have also much more subtle type of appearance, and rarely we can have sneezing and rhinitis. So why all these different manifestations? Why food allergy? Like I tell my students, like if you are dealing with a dog that is extremely weird, you can't pinpoint what the heck is happening in this dog, think about food allergy. Because if it's an allergist, that this is the one that presents with the most variety, the more weird type of presentation. It can go anywhere from an urticaria reaction to a vasculitis-associated presentation. So probably because like we know that food allergy in dogs, well, no is a big word, we think that there is a multifactorial association of immune response. So most of these dogs will be type 1 associated immune response, so more like a immediate IgE response, but we do have a chunk of them that do develop a type 4 hypersensitivity reaction, so more like a delay. In some cases, a type 3 has been hypothesized, specifically in cases in the rare instances in which there is an association with vasculitis or glomerulonephritis, so people were thinking more like immunocomplexes disease that can happen in this particular patient. But by far, most of the dogs will be kind of grouped in a type 1 or a type 4 type of hypersensitivity reaction. And that's come very important when we're talking about how to make a diagnosis of food allergy in our patient. As you guys know, as of today, there is no like a test that actually can confirm a food allergy outside of a very strict, restricted diet. So any type of test is actually not working, is not scientific, is not specific, anything from blood, and I know that you guys have heard of this, but this is my daily practice. Sample from blood, hair, saliva, I mean, you name it, the biological fluid that they test for diagnosis of food allergy, they're all over the place. So still like what we are after is the strict food trial outside of lymphocytic proliferation assays, which for practical reasons are not so diffuse, that they're still just for research purposes. So very important to try to figure out what type of reaction we have in this patient. And in some of those, it's very easy to determine when you challenge them, and you have a owner that says, well, I start to react within 2 minutes. You know, you start to eat the first cables, and boom, like you start to have scratchy dog, or like the nose start to be red again, or you start to develop urticaria, which is a typical type 1 type of hypersensitivity reaction. Obviously, most of our patient doesn't work that way. Otherwise, life would be much easier. So in terms of pathogenesis, when we talk about pathogenesis of food allergy, again, most of those that come from information comes from humans, with some study done in dogs or in mice. And obviously, when we talk about allergies and pathogenic, we cannot avoid to talk about the immune system. So most of this, I'm going to focus on GI. Yes. You heard right, GI from a dermatologist. But there's a lot of similarities between the GI barrier and the skin barrier. Like we saw with Dr. O'Mahony about the skin-gut axis, which imply like a defect in the skin barrier, you get a sensitization, then that reflects as a food allergy after the exposure during when you eat such foods. And that's been shown multiple times in children. Like there's many theories in which associate the food allergy well, because children can, when they eat, essentially they smear the food on their faces when they are kids. If you have an impaired skin barrier, that's the route of exposure. And the little association, little amount of allergen coming through the skin are actually predisposed to an hypersensitivity reaction later in life, hence atopic dermatitis development or food allergy development. The opposite that we'll see better later when we're talking about high amount of food ingested, and that actually trigger the opposite chain reaction, which is more like a protective type of reaction, which is essentially tolerance. So when we talk about GI, there's a huge amount of immune system. We learned yesterday enormous numbers in terms of amount of immune cells present in the skin, in the GI. I would say they are the same amount in the skin. GI is a huge surface area, just like the skin. So we have all these similarities with it. So we start with the epithelial cells. Like on the top of the epithelium, we have a mucus that is very important in protective, putting as a layer between the pathogens and the epithelium underneath. Epithelial cell, intestinal epithelial cells, just like keratinocytes, are not just there as a bricks and avoid the penetration of pathogens, but they are very immunologically active cells. So they produce a lot of cytokines, inflammatory mediators, alarmins, and protective anti-inflammatory mediators. They are bound just like in the skin. We have the tight junctions that regulate the penetration and the absorption of water and nutrients through the different cells. And just like the skin, they produce antimicrobial peptides, better known as host defense peptide, to try to regulate the type of inflammation, as well as try to regulate the proliferation of pathogens. And we have shown a long time ago the same host defense peptide that we find in the skin, we're able to find in the GI of healthy dogs. And we're talking about the beta-defensin and catalycidin, which is the same that Dr. Homan presented in the skin. And then we learn how much is important, like what we eat. We are what we eat. You know, like there was an old advertisement, like, tell me what you eat, I know who you are. It is true, like we are what we eat, and what we eat has a huge impact on the local cell population, local immunity. So in this case, we have like any absorption of vitamin A coming from food. It will stimulate a more regulatory type of immune response, blocking a type 2, and increasing the number of ILC3, so intracellular, intraepithelial lymphoid cells, type 3, which are more regulatory in nature. And the same if we're eating cruciferous, then we increase the ligand for AHR ligands, which has essentially the same effect down the cascade. So we are activating more a Treg type of population, and the Th1 decreasing a Th2 or inflammatory type of mediators. And IL-17 can go up as a protective mechanism. IL-17 are important cells, are inflammatory, but they are important for the defense against micropenetration. And that is even increasing the moment in which we give salt in our food. So salt is not necessarily a good thing, but it can actually act as an inflammatory type of food. Then in the moment in which we have vitamin D exposure, and vitamin D we know is important for a lot of things, but vitamin D, it does stimulate the production again of Tregs at Th1 and Th17. Also very important, the vitamin D does increase host defense peptide. So it does increase the production of this peptide that help to regulate the entire immune system at the gut level. And then finally, we have been learning about fibers and microbiota the entire day yesterday. We will keep hearing about microbiota. We will keep eating about fiber. How important is the fiber in our diet for the production of short-chain fatty acids and other metabolites? And sure enough, here I am showing you exactly the same thing. So when we're eating fiber, specific soluble fiber, we are increasing in the short-chain fatty acid, which do have an immunoregulatory role. Obviously, microbiota, GI microbiota is important, not just for the production of short-chain fatty acid, but they do have a direct effect on activation of dendritic cells, again, the ILC3 type, and the production of IL-10, the retinoic acid, which stimulate, again, the Treg production as an adaptive tolerance. So we start to talk about tolerance. We start to talk about increasing IL-10 regulatory cytokines, increasing TGF-beta in our patient. And that's what was supposed to happen in normal patient. Now, this can be a child, it can be a dog, or can be potentially a cat. More in details, this is a graphic representation of what happened normally in healthy humans to induce tolerance. So you have an exposure of allergen. These get captured by macrophages exposed to dendritic cells who migrate from the lamina propria to the mesenteric lymph nodes, and they do tend to stimulate through the production of TGF-beta T regulatory cells, which start to express the alpha-4 beta-7 chain, which are gut homing receptors. So they migrate back to the intestine, and they tend to behave more as a regulatory cells, inhibiting the activation of even of mast cells. So all of this is good and great. And also we have on this other side that dendritic cells can activate B cell to produce IgA. And we saw yesterday how important as a marker are the IgA in our patient for a good gut function. So that's all what is happening in normal. Obviously, when we have allergy, some of these mechanisms are not functioning. It's not just what it comes through, but how much it comes through is also important. So like the amount of allergens, the amount of antigen coming through the cells, the capturing, is also important in the sense that in normal, again, we're still talking about normal animal here, low antigenic load, they tend to stimulate Treg production, where you have a high antigenic load, then it tends to induce dilation and allergy. So that's the two mechanisms of central tolerance, central peripheric tolerance that we are accustomed to. And if you have high antigenic load that can find the way to the portal vein, then you have a deletion in the liver as well. So tolerance is essential. Tolerance can be driven by different type of mechanisms. So we have low-dose antigenic stimulation, high-dose antigenic stimulation, and both of them end up to have active suppression or clonal deletion and allergy as a mechanism of oral tolerance, which is driven by, again, a multitude of factors. So we have host factor, immune regulatory factor, environmental factor that we listen so far. So it's like all these pieces together that are able to induce a tolerogenic environment in normally. That's why we don't react to every single food that we come in contact with. What happened with food allergy? Well, obviously something went wrong. And this is a very simplistic scheme. I love this picture because I love cartoons. So it's kind of like a very simplistic. So you get exposed the first time, then you get activation of B cells and T cells, and then you start to produce IgE in there, and mast cells and basophils come along. Well, it's a little bit more complicated than this, obviously. So when we start to look more in detail, obviously in this point, we're already to see some disruption of the barrier, just like we see in the skin. The epithelium is not intact. There is a problem. There is like the leaky GI, like everybody has been more and more coming aware of. I'm talking about my specialty. You guys know everything about leaky guts. For us, it's a new concept. So you start to see like a alteration of the barrier. So you have the damage in there. You have increased antigenic uptake. And the production of inflammatory cytokines, inflammatory mediators, the classic IL-25, 33, and TSLP that we saw like a minute ago talking about atopic dermatitis in our patient. When you have this activation of pro-inflammatory cytokines, there is an activation of dendritic cells in the lymph nodes, which stimulate the production of Th2 through the OX40 receptor. So once you have a Th2, then you have all the cascade of pro-inflammatory mediators. You start to have IL-4, IL-13, IL-5, all those wonderful mediators. They are so good, but also very deleterious for our allergies. In all of this, we also start to see the presence of Th9 cells, which end up activating and recruiting mast cells in the GI mucosa, and that's where you have the production and increase in IgE. So in healthy dogs, in healthy humans, we do have an intact epithelium. We have a very good, healthy gut microbiome. We do have a minimal infiltration of inflammatory cells, and then when you start to see more like an allergic patient, you start to have these holes inside the epithelium. You start to see alteration in the microbiome. I'm not going to talk a lot about microbiome because it would be like better speaker than me following me talking about it, and we listened a lot yesterday as well. But essentially, in the moment in which we have alteration of the epithelium, we have a potential for bacterial translocation, and we learned yesterday about the breast milking and the potential translocation from a GI and the activation of more a type 2 type of reaction. And once again, in this very similar graphic highlighting the importance of the leaky gut in the activation of a Th2 type of response, or even increasing capturing of allergen and stimulation of a degranulation of mast cells in the site of the submucosa. So another thing that when I started at the beginning, I was saying is like, well, it is all type 1, type 4. We talk about mast cells. We talk about IgE, which is type 1. Talk about Th2. So which one we're dealing with our patient? And that's when humans medicine become very black and white at times. So we have a specific characterization of food allergy. They are predominantly IgE-mediated, non-IgE-mediated, or mixed IgE, no IgE-mediated. We don't have that classic classification in dogs. We don't have the eosinophilic esophagitis type of presentation specifically associated with the mix, non-IgE-mediated, or we have the anaphylaxis in our patient. Again, they're like the few of them, they do present with this. So how do we answer that question? Well, there is a couple of studies, and they try to help us to define that, the frequency of the immune response. And again, this is a critically appraisal topic by Dr. Olivier and Dr. Mueller, and in which they reviewed the literature asking a specific question. And the way how they were answering the question of type 1, type 4 is, how much time does it take for a dog to relapse, to have a flare of the clinical signs after re-exposure? And essentially, they collected about more than 200 dogs and more than 80 cats. And what they were looking at was essentially when the reoccurrence of the clinical signs appeared after re-challenge. And you can see here essentially that the exposure time to flare, that's its TTF, time to flare 50, so 50% of the dogs flared after 5 days and 4 days in cats, with 90% of the dogs flaring after 14 days and 7 days in cats. So we are looking at quite far away from a classic type 1 hypersensitivity reaction. So people say, well, even type 4, it doesn't take 14 days to develop, right? So what else is going on in this patient? Well, other couple of studies came out to help. This is a prospective study published a few years ago in Japan, in which they essentially enrolled 300 dogs. Out of 300 dogs, 46 were diagnosed with food allergy after a restriction diet with a horse meat-based diet or salmon-based diet. I would never use salmon as a restriction diet, but it worked. So you can see the PVAS, which is the Pruritus Visual Analog Score, and they see after 4 weeks of diet, there is a significant decrease to show that the dogs were doing better. And then they re-exposed these dogs to the previous diet in a very controlled fashion. So owners were to pick when they were at home, and essentially were literally watching their dogs after giving the first cable and see every 30 minutes, 1 hour, 2 hours, how fast the dog were reacting. And essentially what they were able to see was that within the first 12 hours, 61% of these dogs reacted. So they start to have flaring, with about 83% within 1 single day of re-exposure. But we still have some of them that were not reading books, and they took about 17% of them, they were up to 10 days before start to reacting. So once again, it seems that most of these dogs are more a type 1. If I need to classify in a box, I would pick more a type 1 hypersensitivity reaction in these dogs. And by the way, the clinical signs disappeared with the steroids. But what happened again for these guys? So another study very recently published using a amino acid-based diet. They enrolled 62 dogs by dermatologists, and 40% of dogs were diagnosed with food allergy. There was the same thing, same scheme, tried to figure out like how quickly these dogs were relapsing or were flaring, with only one difference. Now we are controlling the amount of food we are giving them. Remember I told you the amount of allergen, the loaded allergen, is also important to trigger a reaction. It's not just when that happens, it's how much do we give them, and this is very known in children as well. So every day they were going up from 1 teaspoon up to 100% of the previous diet, observing if there was any relapse. So when you put the data together, the mean time to flare was 5 days, with 12% of dogs relapsing within 1 day, 88% over 8 days, 7 days. And then we have the one in the tail, 12% of dogs that took up to 14 days to kind of relapse. And the mean amount of volume needed, it was 50% of the previous diet. So with very few dogs reacting at 1 teaspoon, so after the first day, and one dog needed 1 tablespoon, so reacting on the second day. So definitely the amount of food, it does have an effect on how fast this dog flare. And that's what we see in practice, right? We never say, oh, go back and give it the same diet, like relapse. You give it the 100% of the previous diet. You're going to see diarrhea. Of course you do. But most of the time we say, well, change it back, like 1 a day, 2 a day, and keep going. So faster you do that, that's when you reach like a critical point and they start to develop allergies, like the relapse. So Bottom line is we still have a type 1, most of them. We do have an important type 4, though, and we don't need to forget, and the amount of allergen that this dog get exposed. So in terms of allergens, when we're talking about allergens are important. They are not all created equal. Those are the 9 major food allergens in children, with 25% of children be allergic to peanuts, going down to soy in about 5%. And even when we're talking the same allergen, many food have different type of allergens. Those are just kind of examples. Everybody's familiar with peanut allergy in children, and the different RH1, 2, and 3 as a major allergens. And then we have the eggs, which there's some important egg allergen, which is this guy right here, which is the equivalent of, as a very high homology for the serum albumin in chicken, and we'll come back later. And then also the cow milk allergy, the difference with the goat milk is just the amount of the type of casein chain that is present in this diet. So what are the allergens in our patients? Well, very biased studies done over the times. I say biased because it depends really what the people decide to challenge these dogs with. So if I'm starting with beef, obviously the beef comes as the most common allergen. If I try to decide with, if I'm going with soy, it may be later on, or, that's what I mean as a biased. And over the time, allergen change. So you see, for example, chicken, it went from 9% to 15% after about 10 years of reviewing a new paper coming out. And we did the same for cats, in which from beef it became lamb, the most common allergen, just because of the increase in lamb ingredients in the diets over the time. So that's why I'm saying take this with very much biased thing, but definitely beef, chicken, dairy product, those are still the most common allergens that we see in our dogs and cats. So I said, no, all the allergens are created equal. They do have specific characteristics that they need to respond to. They are generally protein or glycoprotein. They have a specific molecular weight, and they are thermoresistant, hydrophilic, resistant to acid degradation. Well, all of these make all sense, right? They need to go to the GI, they need to be absorbed to the gut, so they need to pass the stomach and the digestive enzymes. So those are the classic characteristics of an allergen. And you see that also the important is how the allergen gets processed from the native form to the one that is actually exposing into the GI. And I said they're mainly proteins, but the proteins are associated with something else. So can be associated with lipids or can be associated with sugar. And lipids are actually very important in changing the allergenicity of these allergens. So maybe delay the degradation, or they may expose or hide specific allergenic epitopes, or even increase the absorption of this particular allergen. So the binding, like the lipoproteins or glycoproteins, are very important. And in terms of glycoproteins, obviously the association between proteins and carbohydrates, we need to remember the Maillard reaction, which is something great for people that eat meat or vegetables, but it's also the production of Maillard reactive product, which are not necessarily good for the immune system. So association between proteins and sugar with high heat produce these reactions. And essentially, the Maillard reactive product can induce different type of immunological mechanisms. Most of the time, though, they do tend to produce an inflammatory type of reaction. They tend to stimulate more a Th17 and Th2 instead than activated Treg and the Th1. So, but that's a kind of something that we need to deal with. Like Maillard reaction are good for flavored aroma and for eating purposes, like to incentivize to eat. On the other side, though, we need to kind of take in consideration the amount of inflammation that this product may trigger in our patient. Not a lot of allergens have been characterized in our patients. There's only few studies showing allergens from fish and chicken, and mainly are all serological studies. So they're all in vitro studies showing the most common allergies specifically looking IgE type of expression. And this is a study back in 2018 in which they were able to identify 9 different types of allergens, which, by the way, are also common proteins and allergenic substances found in people. And they are commonly identified in chicken, in fish, and crustaceans. Well, chicken and fish, they're not really the same, but they do have the same proteins inside, and that's what we are recognizing it. So when we're looking about chicken allergens, again, like another serological study, taking consideration sensitized dogs to chicken, non-sensitized dogs, and allergic dogs to chicken. And they were able to show essentially the presence of 7 major allergens. And you can see the one in red, so the serum albumin, the enolase 3, the creatine kinase M, were present in every single allergic dogs and sensitized dogs. Instead, like in yellow, highlighted in yellow, you will see the sequence, and they were homologous for the fish type of protein. So there's a lot of commonality between fish and chicken, so mammalian and birds and fish, which complicates our life quite a bit. And they also were able to identify the minor allergens. So just to refresh, major is when you have 50% or more of your population reacting to something. Less than 50% is considered a minor allergens, which in this case it was troponin C. And this paper also brought up the concept of pan-allergens as an important concept in our allergic patients. So pan-allergens are those allergens that have high homology across the board. And you can see in this study in which, for example, duck and turkey, you have high homology of about 90%. So essentially you have 90% chance that if you are allergic to a duck and you feed the turkey, you may be reacting to the turkey. So some of those are due to albumins. That's why I said it's important in the chicken, were important in the serum albumin in this study. So albumin are considered panallergens because they are homologous in many, many species, growing across from mammalians to fish to birds. And essentially you have dogs that are allergic to chicken, but they can react to alligator just because they share high homology in the serum albumin. So that makes a huge deal when we're dealing with how do you make a diagnosis of food allergy, identify novel protein diets. Well, it's a problem. So, and that's the problem. You may need to be very careful when you select the protein that you really want to, the source of the new protein for our patient. And to make it more complicated, there's a lot of going on in terms of cross-reactions between different proteins. And there's, again, like a few studies, again, serological study, showing that, just for question of time, I'm running a little bit through this, but essentially we have a high cross-reactivity between lamb, beef, and milk in our patients. So they tend to go together because they are kind of closely related to each other. And in fact, one study that I will show later, they look specifically at the amount of of ruminants, how much the ruminants meat cross-react with each other. So when we're looking at chicken, this is another study done in dogs and cats in which they were able to show essentially that chicken, duck, and turkey completely cross-react with each other. So this is an inhibitory assay. Essentially, the plates were coated with chicken, duck, or turkey, and then they were IgE against, in this case, like the 3 meats, so the turkey, the chicken, and the duck, and see how much inhibition you got. And it was anywhere between 85 and 90%, independently from what it was the substrate coating. So when they had the internal control, like the second one, the BMT is the beef, is supposed to be the negative control. And then they're looking for non-hydrolyzed chicken meal, hydrolyzed, moderately hydrolyzed chicken meal, and then extensively hydrolyzed chicken feathers. And then you see that the extensive hydrolyzed, it was the one with the lowest amount of reactivity of inhibition, below the 5-10%, to show essentially that if you want to inhibit that type of cross-reactivity, you really need to go below, in terms of chicken, below a 10 kilodalton type of sites of proteins. And this is essentially the same study done by the same group, but looking at this point, the cross-reactivity between whitefish, salmon, and chicken. And also in this case, they were able to show a very strong inhibition, aka cross-reactivity between these three types of foods. Sorry, I'm running a little bit because I'm running out of time. But when they're looking at correlation between ontogenically related species, that's when you start to look at, there's a major correlation between ruminant allergens, specifically beef and lamb, or like grains allergens, soy and grain. In US, I learned that from a student many years ago, that soy is actually considered a grain. So when people consider like a grain-free diet, and that they do better, maybe it's not because it's the grain, but maybe because the dog is allergic to soy, and you are not having soy exposure. So it depends, but also again, like poultry allergens, they cross-react with each other in terms of chicken and duck, for example. So more closely related they are, and more likelihood you have that they're going to react to each other. Once again, the problem with this type of study is they're all IgE mediated, are all laboratory. There are no good studies showing the in vivo exposure and reacutization when you have a different allergen exposure. The only one that has been actually shown in vivo is the chicken and duck. So there were chicken allergic dogs exposed to duck that do relapse. Most of those dogs do relapse clinically, but all the others are all essentially in vitro studies. So we do need more in vivo to confirm the true evidence of this cross-reactivity that we see in vitro. So in conclusion, we know a lot about pathogenesis. We know everything about pathogenesis of food allergy, clearly. Joke. So we don't know very little we know about that. There is a variable clinical presentation, depending also of which specialty you are talking to, and the importance of different type of hypersensitivity reaction involved in different patients. So every dog is a unique when we're talking about food allergy. And then obviously all the complexity of the allergens, pan-allergens, and the physical chemical structures of the allergens, how that is important. It's not just the size, but also how they react and which allergen we're actually talking about for our patient. And with that, I want to just to thank you guys for your attention. Maybe guys, most of you are not familiar with that. That's Florida, so we do have a lot of alligators, and the gator is a symbol of UF. So that's why the gator.